Welcome students to the laboratory demonstration. For the laboratory one, students need to do two experiments. The first one is stick plate, and the next one is gram staining. First, students will get one tube of the unknown sample. This is a mixed bacterial culture, so it means this tube contains one type of bacteria. And students need to use the bacterial suspension in this tube to streak onto nutrient agar, blood agar, and microalgae agar. And then also use bacterial suspension for gram staining as well. For the gram staining, students will do the gram staining of Escherichia coli and Staphylococcus aureus, which are the colony on the agar plate. These two types of bacteria can be used as control organism. For Escherichia coli, this is a gram-negative bacilli. So it means after finish gram staining and observe under a microscope, you need to observe a red color. And for Staphylococcus aureus, this is a gram-positive cocci in cluster. So it means you need to observe the purple color if you do it correctly. In laboratory, we are going to use heat to sterilize. So students need to learn how to use the gas tank. This is a gas tank in the lab. First, you need to open the door and turn on the valve with a counterclockwise direction and press a small button like this. After that, you can observe the movement of the pointer. And put it back, close the door, and sign your name, which including your name and experiment date on a logbook. After that, students need to turn on the yellow valve on the lab bench as well, and also the valve of the burner as well, and use a gas lighter. After finish your experiment, you need to turn off all valves on your table and the gas tank as well. You can turn off with a clockwise direction. Put it back, close the door, and sign your name. Let's start with the first experiment, stick plate. Student will get a mixed culture tubes and three plates, which including nutrient agar, blood agar, and microalgae agar. And also get two loops as well. First, you need to label on the plate, which including your name, the number of unknown sample, and experiment date. Because after this, all plates need to incubate together. So that's why you need to label your name on each plate. After that, you need to mix the culture by vortexing the tube and open the cap. After that, pass the tube to a frame This is a sterile technique and use the loop into the bacterial suspension. You can observe a thin film layer in the loop like this. Then start with the nutrient agar plate. So start the first quadrant like this. After that, you need to frame the loop and let it cool down. You can use the another loop and drop the smear from the first quadrant to do the second one and frame the loop again and continue the next quadrant. So we're going to do 4 quadrants for each page. So don't forget to frame your loop before doing the next quadrant. And the next one, blood agar. You can do the same. Start the first quadrant, frame your loop, and continue the second. And then continue the third. Frame your loop and continue the last quadrant. When you finish the last quadrant, you need to step your loop 3 to 4 steps onto a free area because you need to observe the hemolysis patterns, which including beta, alpha, and gamma hemolysis. And next, you're going to do with the McConkey agar as well. For the McConkey agar, you can observe the lactose fermentation of bacteria. After you finish this, and the plate will be incubated, and the next day you can observe a single colony on each plate. Okay, and next experiment, the the game standing. First, you will get to control bacterial agar paste, which including Escherichia coli and Staphylococcus aureus. And you need to use the normal slide as well for the bacterial colony. And the next slide you need to do with the mixed culture tubes or the bacterial suspension. So it means you will have two glass slides. The first slide, you can use the sample from two agar plates. And the second slide, you use the sample from the liquid media culture or the mixed sample. First, before preparing the smear, you need to clean your glass surface first. You need to clean with Scotch and 95% ethanol. You need to clean it thoroughly. You need to clean the surface like this. You need to clean both sides of your slide. After that, you need to flip the slide and label the name of microorganism. For the first slide, you need to label Escherichia coli and Staphylococcus aureus. You can do like a half. The left side is Escherichia coli and the right side is Staphylococcus aureus. And then you can draw a circle area for bacterial smear. Like this. And the next slide for bacterial suspensions or the mixed culture. You need to label the name and draw a circle area as well. For samples for mega, you need to drop the normal slide. You drop outside the circle area first, and then use loop. Touch the normal slide and touch into the circle area like this. Just only one loop. If you use more than one loop, this means you need more time to dry your slide. And then you need to pick a colony, just a tiny. And then smear into the normal slide. You need to smear to spread the sample. And do the same with Staphylococcus aureus. And let it dry. You can observe the smear area like this. And next, The preparation for bacterial suspension. You need to mix the culture first and then use 2 to 3 loops of bacterial suspension. Tap onto the circle area and then smear to spread the sample into a circle area. And let it dry. And next is a fixed step. Students need to pass the slides through the frame. You can use the faucet and do 2 to 3 times. Not frame it, just pass through. Okay, we are going to start with the gram staining steps. We're going to use the first dye, which is a crystal violet. The next one, glamazine, and 95% ethanol. And the counter stain, which is a saphonin. First, you need to bring your slides through a sink and place them like this. After that, you need to drop your first dye, which is a crystal violet. Drop on a circle area, not for full area, just a circle area. And then let it stain for 1 minute. After that, pass the tap water through the slide. And then you need to drop gramazine into a circle area as well. And let it stain for 1 minute as well. And then pass the tap water through the slide. The next step is decolorization. And use the 95% ethanol. You need to decolorize approximately 5 to 10 minutes. It depends on your preparation. If you prepare thick layer of your sample, you may need more time to decolorize. So you need to decolorize until it shows no purple color. And after that, add saphonin and let it stain for 30 seconds. And after that, pass the slide to the tap borders to remove the dye. After that, use dye blotting paper like this, and then tap the paper on a slide surface. Please gently tap and do not break the glass slide. And after that, you can bring your slides to observe under a microscope. So for Thursday and Friday, you are going to do 2 experiments. The first one is gram staining, and the first one is a stick plate, and the next one is gram staining. So see you on Thursday.